Hello, and welcome to Cognitum, a show dedicated to exploring the present and future of science and technology. I'm your host, Iosif Gershteyn. Today with us is Dr. Leonardo Ferreira. Leo is an immunologist at the University of California, San Francisco, at the Tang and Bluestone Labs, and a founding fellow at the Ajax Biomedical Institute. Leo, welcome. Tell us about what you're working on now.
My work right now focuses on using immune cells as living drugs.
Could you explain what that means?
So right now, medicine focuses a lot on treating symptoms, when then you could imagine there's two pillars. One using small molecules, such as aspirin. A second one, more recent one, is using antibodies. So proteins that can recognize other proteins, and therefore they can neutralize their activity. I think there should be a third pillar of medicine, that we can actually use cells as living drugs. Cells are much more complex entities than small molecules or antibodies. And so you can really program these cells to go where you want, when you want, and do what you want.
And what would be the key differentiator of this versus the other two pillars you mentioned?
So I think one difference will be the degree of control that we can have. If you take an aspirin, you'll just go in your bloodstream and go everywhere in your body. The same thing with an antibody. You're going to have to inject IV intravenously this antibody. It'll just go everywhere in your body. If you have a cell, however, you can imagine programming that cell to go only to a very specific location in your body.
And what would be the benefit of going to a specific place instead of systemic effect?
There's many conditions that would greatly benefit from having a local effect versus a whole body wide one. And one example is of autoimmune disease. In autoimmune disease, your immune system is recognizing and killing a specific cell type in your body. It's like the body at war with itself. For example, in type 1 diabetes, some of your immune cells, your T cells, will recognize your pancreatic beta cells, the ones that make insulin to control your blood sugar levels. And so if you look into the pancreas of these type 1 diabetes patients, there's a full-blown immune response going on there. So if you could design cells that are programmed to go specifically to the pancreas and put off that fire, that could be potentially a lifelong therapy.
And this is what you're working on?
Correct.
So how do you create these engineered cells?
So the technology I'm focusing on right now was actually first used in cancer. They're called Chimeric Antigen Receptors, or CARs for short. So these CARs are chimeric molecules because they're made of two components. One component stays outside, on the outside part of the cell. It's actually based on an antibody. So it can be designed to recognize any cell surface molecule you want. Imagine you find a cancer that expresses a molecule on its surface, a protein, that is not found in healthy cells. You'd want to target that molecule and not some other aspect that is shared with other cells. So that's the outside. The inside part of this molecule is an array of signaling modules. And cell signaling is how cells make decisions. And so if you can engineer that signaling domain, you can basically control how this cell behaves. And so when you put these two parts together, you have a CAR. And this CAR has really been a game changer for cancer therapy, because you can now you can build this receptor custom-made for a specific cancer and to elicit specific responses. So the most successful therapy right now is the CD19 CAR. CD19 is a receptor that is expressed in all B-cell derived leukemias.
So CD19 is a receptor, which is, for our intents and purposes, we could call it a marker.
Correct.
that's only on the cells that are cancerous with this type of cancer, but not in healthy cells.
So it's only on B cells. So in this specific case, all B cells express CD19. And so B cells that become cancerous, become leukemia, they now also express CD19.
Mm-hmm.
And other cells in your body do not express this gene.
So leukemia-affected B cells express CD19.
Correct.
And healthy B cells do not express.
They do.
They do. So how does the cell that the CAR, affect the cancerous cells and not affect the healthy cells?
So in this case, this therapy actually kills off all your B cells,
Mm-hmm.
whether they're healthy or cancerous. But it does not, in theory, affect any other cells in your body. And it is possible to live without B cells. It's not ideal, but it's better than being killed by the cancer, because an acute leukemia used to be a death sentence up until the development of this technology. And so this is an example, although not perfect, of a molecule that is expressed only in a subset of cells. And so you can basically rewire a T cell that naturally doesn't see it to go and destroy these cancerous cells.
But it's not specific enough to actually only destroy the cancerous cells. It destroys the whole class of cells.
Correct. All B cells are wiped out in these therapies.
Mm-hmm.
But there's many patients that would have died otherwise and are now still living because of this therapy. Because we were able to take their T cells out from their peripheral blood, we engineer them to express this CAR receptor against CD19, and then put them back in. And now these T cells are ready to attack. And anytime they find a CD19 expressing cell, including all the cancer cells, they will just destroy them.
So the T cells, which are immune cells, which could you give a little bit more background on T cells before we get into
Yes,
that?
so T cells are adaptive immune cells. They have a T cell receptor, which is very special because this T cell receptor allows them, the T cells, to see inside cells. Because the T cell receptor evolved to recognize peptides, so protein fragments, presented by a specific surface molecule called the MHC, the major histocompatibility complex. And so you have a cell that's going at its normal business, and so inside the cell you have the instructions book, the DNA, which makes a message called RNA, which then gets translated to produce proteins, which are the machines inside the cells. But these proteins don't last forever. They have to be degraded at some point. And so when they're degraded, they become peptides. So peptides are just pieces of proteins.
Degraded means broken down.
Correct.
So it's like if we use the analogy of a car, it's taking it to the dump, breaking it apart into parts, and then throwing out the parts.
Correct.
And the parts would be the peptides.
The peptides.
Mm-hmm.
Yeah.
And they're thrown out outside of the cell wall.
Not so fast, because they actually stay inside the cell.
Mm-hmm.
And some of these peptides have just the right structure to bind to these MHC molecules inside the cell. And now this complex of the protein piece, the peptide, and the MHC, it goes to the surface of the cell. It's basically like they're holding an ID card and say, hey, this is what my proteins look like. And so the T cell, which again, it can't see the surface of the cell. The T cell can only see peptides that are presented on MHC molecules, goes and senses it. And then if it's a self peptide, if it's your own protein, a normal protein, your T cells let it go. Because the T cells went to school in the thymus and they learned how to tell self from non-self. So if it's just your own protein, unmodified, it's okay. But if your cell is infected with a virus, or if it's becoming cancerous, it will now have modified proteins. And your T cells, or some of your T cells, will have just the right T cell receptor to recognize those modified versions of proteins. It's like you have a cell and it has windows, and so the window is open, so the T cell can peek into that window. And now it can tell, the T cells as a whole population, they can tell if that's a normal protein or if it's a mutant protein or a foreign protein from a virus, for example.
And if they identify it as a foreign or mutant protein, what do they do?
So if it's a cytotoxic T cell, a CD8 T cell, it will kill that cell. And this is why many viruses and many cancer types, they actually somehow learned to downregulate, to bring down the levels of MHC on the surface in the hope of becoming invisible to the immune system. But even then, they don't succeed, because then there's another immune cell type called natural killer cells, which, as their name indicates, their standby mode is on. They were born to kill. So if this cell now loses MHC on its surface, it's as if it was closing the window. And so if you have a cell with its windows all closed, the natural killer cell goes and kills it. It's called the missing self hypothesis, in which natural killer cells will kill any cell in your body that is missing self.
Mm-hmm. And how does this relate to what you're trying to accomplish at your lab?
So in this case, even back to the cars, these cars really expand the horizon of what a T cell can see. A normal T cell can only see inside cells, can only see peptides presented by specific MHC molecules. If you have a CAR receptor, you can rewire a T cell to see anything you want, because the CAR will determine what the T cell responds to and how it responds to it. So in cancer, the idea was, let's take a molecule that is as specific as possible to this cancer cell type, and then rewire the T cells to recognize and kill it. In my case, I'm actually trying to use this technology to induce immune tolerance.
Mm-hmm. And what is immune tolerance?
So immune tolerance is a very special state of the immune system where it becomes unresponsive to a particular cell or organ that could invoke an immune response. And the best example is actually transplantation. So a body of healthy individual, the immune system does not recognize your own cells. And that's immune tolerance. Because if I am to transplant an organ to someone who is not matched with me, that organ invokes an immune response in the recipient. Therefore, you have a state where my immune system is unresponsive to my pancreas, for example, even though my pancreas, in theory, could invoke an immune response if you were put elsewhere.
Mm-hmm. So your immune system knows not to attack yourself.
Correct.
But when you transplant a foreign body inside your body, then the immune system will naturally attack that foreign body because it is not yourself.
Yes, and this is what we call immune rejection,
Mm-hmm.
which is a phenomenon then where you transplant an organ into someone who's not genetically matched with you, that organ will be rejected. And this is very problematic in the field, because imagine you have a terrible accident and you need an organ transplant to survive. You have not only to find a healthy liver or kidney or pancreas, it also has to be matched to you. But this is very hard to accomplish, because these MHC molecules that I mentioned, they are the main drivers of immune rejection, of self, non-self recognition between different people's immune systems. And there's many flavors to each one of them. Just for MHC class 1, which is one group of MHC genes, there's HLA-A, HLA-B, HLA-C. HLA-A, this gene alone, has more than 2,000 different flavors. So it's almost mathematically impossible to find two people that have the same exact combination of alleles. So usually you try to go to a family, to a relative, or to someone across the world, but they have to be matched with you at the MHC level. And so actually many people die waiting for an organ. In the US alone, it's more than 10,000 people, or like 20 airplane crashes, if you will.
20 airplane crashes
every year.
A year of people just dying waiting for an organ transplant.
Correct.
And this is all based on the problem that there's a lot of diversity in MHC, or people's genetics, and so the body will reject an organ that's not perfectly matched, and it's very, very hard to find a good match.
Yes, which is not a bad thing from the evolution standpoint, because evolution shaped us as a population, the human race, to have very diverse MHC. And why? Because if there's some virus, some plague that affects us all, just by chance, a few people will have an MHC variant that is very good, at presenting peptides from this virus, and then has a very good T cell that will kill those cells. So any virus that comes at us, we as a whole race will survive. But, of course, evolution did not think of organ transplant. That's not what we did like in prehistory and even before that. So now a modern problem of this diversity is that it's very hard to find a match for an organ transplant. And what I'm working on is thinking about, like, what if it didn't have to be that way? What if we can re-educate a person's immune system to take an organ from someone else?
And how would we do that?
So the current idea that I'm working on is to use immune cells to achieve this tolerance. So luckily for us, not all T cells are meant to kill. Some T cells actually regulate the killing of other T cells. They're a very rare subset group of T cells called regulatory T cells, or TREGs for short, as we call them. And so if you see the immune system as an army that can be mounted to defend us from invaders or from a transplant organ, which again is not bad for us, but your immune system doesn't know. You can imagine these cells to be the generals of that immune system, because they make sure that nothing goes overboard. So if you're fighting a microbe, you have to mount an immune response. But at some point, when the microbe is done, you really like eradicated that microbe, you want the immune response to go away. So it should go up and then back down. Tregs are at work there. They make sure that once the microbe has been eradicated, your immune system comes back down.
So the regulatory T cells are the generals of this T cell army.
Correct.
And what do you do with these regulatory T cells?
So what we're doing right now is we can purify them, so we get blood from healthy individuals, from type 1 diabetes patients and other autoimmune disorders, and you can isolate, purify these cells using flow cytometry. So this is one technique where you can take immune cells, you can label them with different antibodies with different colors on the surface, and then you pass them through this machine which has a system of lasers that can read cells, just like a barcode. If you go to the supermarket and you want to scan your milk and your cereals, the machine can tell which one is the milk, which one is the cereal. So likewise, this machine can tell which ones are cytotoxic T cells, which ones are natural killer cells, which ones are regulatory T cells. And then more than that, you can actually separate them in individual tubes. So I can get a whole bag of blood, and I can prepare it such that at the end I have a tube in my hand with only the regulatory T cells inside it. And once I have these cells, I can now modify their genome to introduce a chimeric antigen receptor, but not for killing. I want this chimeric antigen receptor to direct these Tregs to the site of action in autoimmune disease, for example, in type 1 diabetes. I want to engineer these Tregs to go to the pancreas, and once they get there, if they can put out the fire that is happening at the pancreas, if they can inhibit the T cell mediated killing of beta cells that is happening in the pancreas.
So your approach is to re-educate these generals so they go out to the place where you need them. That in your case, the beta cells of the insulin producing beta cells of the pancreas, and direct the T cells not to kill.
Correct. Specific location, specific function, specific timing. And again, because blood has been at the forefront of all these cell therapy approaches, starting with bone marrow transplant. Bone marrow transplant has been happening for decades, and that was the first cell therapy. And now fast forward all these decades, and we can not just put cells in, we can genetically modify the cells before putting them in. And we can actually do it from the same person. So we can take your own cells, grow them in the lab, genetically modify them, and put them back in. In this case, the modification will be to deploy, to kind of teach them, program them to go to this specific location, the pancreas, and be there and like try to stop the immune response that is happening, that should be happening.
Do the engineered cells regenerate? Are they self-sustaining, or do they need to be continually infused?
So this is a very good question. It is now in trials. So the first trial that we did at UCSF was to see if they're safe. And so we found them to be safe, and but their longevity is not clear because we actually label the cells with deuterium, So it's just an isoform of hydrogen that is heavier. So the atom itself is heavier, so you can tell it apart. And so with this labeling system, we find that when we inject the cells, we can detect them at the beginning in the blood of the patients, but then they slowly phase off. And there's two possible explanations. One is the cells are dying. Once they go in, they die. Second explanation is they're homing to the pancreas. And once they're there, they're not coming back to the blood. So this is something that it's not clear yet because it's not as easy to get a piece from the pancreas as it is to get a blood sample. But so the idea is to see how long do these cells live for in the body once they're injected, and can we actually modify that? So I feel like with Tregs, I like to think there is 3 S's that are important: survival, suppression, and stability.
Could you go through each one of them and explain why they're important?
Yeah, so survival is perhaps one of the simplest ones to understand, because if your cells are not alive, they don't really work. And you could imagine that these cells go through quite some stress. You take them out of the body, you grow them in these artificial conditions in a laboratory, you modify their genome, you grow them again, and then you put them inside the person. So you could imagine it'll be more fragile than cells that never left your body. And so if we can actually introduce in this receptor, this CAR receptor, signals that improve cell survival, that's actually a big focus of mine right now. Because what we observe is that these cells, again, because they're more fragile, you can lose more cells than you would otherwise with this receptor. So that will be the first goal, to have them live for long enough to induce immune tolerance. And it doesn't have to be for too long, because the way tolerance works, or should work, is actually an infectious mechanism. It's called infectious tolerance. So you put these Tregs in, they go to the place of action, and they should be able, not just themselves, to stop the immune response, but actually to re-educate the immune cells at that site, so that your own Tregs, the unmodified Tregs, should also learn to become tolerant of this tissue. But they still have to live for long enough. So that's the first S,
and
survival.
roughly what time frame are we talking about when it to re-educate the endogenous and unmodified T regulatory cells?
This is another good question, because I should say that most of the field has done a lot of work in mice, which are much short-lived than us. So whatever we see in the mouse, it's not clear if we have to multiply that by some factor in humans, or if it's the same amount of time. But from these clinical trials, it seems that it will be possible to, within a reasonable time frame of survival of the cells, they can re-educate the immune system. But you can imagine that a T-cell response takes 1, 2, 3 weeks to mount. That's how long it takes an adaptive immune response. Because first you have the innate, so the first line of defense, and then they make cytokines, so they make signaling molecules that call into action other members of the army, the T cells. So when the T cells come in, which takes again a couple weeks, then it can mount the immune response. So then maybe you need some more weeks to re-educate, retrain those T cells once they're there to stop the immune response. Maybe it will take months. That's not clear right now.
Mm-hmm. So on to the second S.
So then the second S would be suppression. So the goal of putting these cells in is that they suppress the immune response. But not all Tregs are equal. There's actually within Tregs, which are a subset of T cells, there's subsets of Tregs. And we immunologists just love it, that cells can be split into types, which in turn can be split into more types. But so for Tregs specifically, it seems that Tregs can specialize further into their job. So some Tregs are very good at suppressing some kinds of inflammation, but not others, and differently for other types. So once again, because you have this CAR, you have a large degree of control over the cell decision-making process. So
CAR is the mechanism that you use to re-educate the T regulatory
Correct.
cell.
So CAR is the chimeric antigen receptor. So we have this artificial gene, so we don't have to limit ourselves to natural occurring genes. We can just make up our own gene. And this gene, the chimeric antigen receptor, really has a signaling component that allows you to program in what you want the cell to do once it gets there. And so you can now imagine that if you have a type 1 diabetic pancreas, it's a specific type of inflammation that will be qualitatively different from other types of inflammation. So you want to maximize the Treg potency to suppress that type of inflammation. So that will be the second S, that the suppression that you are teaching programming these cells to do is the most adequate one for a specific application.
Mm-hmm. And the third S?
So the third S is stability. And this actually pertains to Treg biology itself. So it turns out that regulatory T cells, they're a separate lineage of T cells. They express a master transcription factor, so it's a protein that controls how genes get expressed inside cells. And this gene is called FOXP3. So regulatory T cells are FOXP3 positive. They express FOXP3, which turns on a whole program inside the cell that tells it to be a Treg. But it turns out that this program is not as stable as we first thought. So if you put regulatory T cells under extreme inflammatory or stress conditions, these regulatory T cells can actually become conventional T cells, killer T cells, helper T cells. So once again, in our artificial receptor, we need to make sure that we include the right signals, the right instructions to prolong Treg stability as much as possible. Because this has been a big challenge actually in implementing unmodified Treg therapy. Because in this case, there are some antigen-specific Tregs. So an antigen is a piece of a protein that is immunogenic and is recognized by a T cell. We call it antigen. And so your Tregs are obviously antigen-specific because they have a T cell receptor, just like any T cell. But they're very rare. They're so rare that for you to have a quantity of Tregs they'll be meaningful for therapy, you'd have to expand them massively. And by the time you expanded them enough to have enough cells, they might no longer be Tregs. Because the more they divide, the more likely it is that something will disturb their delicate program. So they're no longer suppressive regulatory T cells.
So if you get all of these three S's within the proper parameters, where do you see this therapy going?
So I think the first application of this therapy will be immune rejection in transplant. Why? Because there we know exactly what we should target. We should target the MHC molecules. Say the example of HLA-A. One allele of HLA-A is called HLA-A2. So if you take an organ from an HLA-A2 positive person, which is like 30% of the population, and put into someone with HLA-A2 negative, it will be immediately recognized as foreign. That person's immune system knows we don't have HLA-A2 here. So if this organ has HLA-A2 on its surface, it can't be ours. We must reject it. We must kill off this organ. What if I were to co-transplant this organ with modified regulatory T cells with a chimeric antigen receptor that recognizes HLA-A2? Or maybe I actually want to give these Tregs before the organ transplant, so the timing issue you brought before. Maybe I actually want to first prepare the recipient of the organ to be tolerant of HLA-A2 expressing organs. And then you put in the organ. This patient has now been prepared, has been re-educated, rewired the immune system to accept his organ as if it was his or her own. And so this would make a huge difference in the lives of, again, 22 plane crashes a year worth of people. Because now you can, in theory, take any organ, regardless of MHC matching, and give to the people in need.
Well, that is fascinating work, and I really hope that we see its fruits very soon. Thank you for joining us this evening. We hope you join us again next time, where we explore the frontiers of science and technology. Have a good evening.